The overall goal of this procedure is to functionally reconstitute mescal channels into a droplet interface bilayer system and to examine the response of these mechanosensitive channels to harmonic mechanical stimuli.
This method enables the study of the mechanoelectrical activity of the V23T mescal mutant in response to harmonic mechanical excitation after reconstitution into a droplet interface bilayer system. The main advantage of this technique is that it utilizes the technical advantages of both the patch clamp technique and the droplet interface bilayer methods, with the ability to be miniaturized as well as the ability to control the chemical composition of each side of the membrane.
To formulate liposomes, prepare 10 milliliters of a 2 milligram per milliliter lipid solution by adding 10 milliliters of potassium chloride buffer to 20 milligrams of DPHPC synthetic lipids purchased as lyophilized powder. Make sure both lipid vehicles and buffer solution are thoroughly mixed. The mixture should look homogeneous and hazy when everything is dissolved. Freeze the mixture at -20 degrees Celsius. Now, completely thaw the new lipid mixture, letting the mixture thaw at room temperature and not in a heated environment. After a total of 6 freeze-thaw cycles, use a commercially available extruder to extrude the lipids by forcing the whole lipid suspension first through a 0.4 micron polycarbonate membrane filter, and then 6 times through a 0.1 micron membrane filter. This process yields particles with diameters near 100 nanometers.